I'll live up to that. This is actually, I guess you could say, what I did this summer, so for my summer vacation. The Arlington Archosaur site is interesting not only for the fossils that we find there, but also the history of the site and how we've been working with the community to get this science done. So we'll unpackage some of this just to make sure we're all on the same page first. These are fossils from the Cretaceous time period. That's the last chunk of the age of dinosaurs. Runs from 145 to 65.5 million years ago. Excuse me. And a lot of our favorites, the names that all little 4-year-olds know, come from the Cretaceous. So things like Tyrannosaurus rex, things like Triceratops. Our own, mascot outside of the McClung Museum at Montosaurus is a Cretaceous species. But what a lot of people sometimes don't know, and what we as paleontologists don't always communicate very well, is our fossil record is actually really biased. And we don't have wonderful sampling across the entirety of the Cretaceous. And to just give you an example of that, this is a chart showing diversity of tetrapods, so vertebrates that at least ancestrally had arms and legs, through all of the Cretaceous. We have this little blip where we have quite a few things known from the Albian, and then a rather huge spike in the very end for the Campanian and the Maastrichtian. But everywhere in between, we don't actually have that much information. We just haven't found a lot of fossils from North America from these chunks of time. So they're a little bit mysterious. We really want to fill in those gaps, though, because there's some interesting things going on, particularly in North America during that chunk of time. So, all right, what about that older piece of the Cretaceous, about 100 to 113 million years ago? This is just an artist's reconstruction of what the landscape might have looked like at this point in time. And who are our main players? Who do we find at sites from this age? Tetnurans, that's we'll say a fancy name for some of these theropod dinosaurs, walk on two legs, eat meat, things like Acrocanthosaurus in this picture. You might have heard the name Allosaurus, they're sort of close cousins to that. Large dromaeosaurs. You've seen Jurassic Park, the velociraptors in the movie. Real velociraptors were about that big, which might be tragic for some of you to know, but there were close relatives that were about the right size, things like Utahraptor here. Members of Iguanodontia, so these are plant-eating animals that have big thumb spikes. We have ankylosaurs, which are these really heavily armored, almost tank-like herbivorous dinosaurs as well. Of course, we have crocs, they're near and dear to my heart. There are some really large, what we call Neosuchians, distant relatives of the ones that are around today. And we get early members of the cryptodire turtles. So cryptodires are the ones that can pull their head back into their shell, kind of in an S shape, instead of pleurodires where they stick their head over to the side. At the end of the Cretaceous, we also have really, really good sampling, and here's another artist's reconstruction of what North America kind of looked like during that chunk of time. And the players in this case are different. We're not looking at the same groups anymore. So instead of having things like that Acrocanthosaurus, we have Tyrannosaurs, not just Tyrannosaurus rex, but several others. We do still have these dromaeosaurs, the guys with the interesting little killing claw on their feet, but we don't have large-bodied ones anymore. They're all kind of small. Iguanodons are out, and instead we have hadrosaurs, duck-billed dinosaurs, like Edmontosaurus here. We also have a lot of ceratopsians. So the most famous one of those would be Triceratops, but there's several other groups of these frilled, horned, herbivorous dinosaurs. We have ankylosaurs, but at this point, these are the ones with the clubs on the tail. So a solid chunk of bone looks about the size of an NFL football. They would use to whack things that the earlier ones didn't have. And Eusuchian crocodilians, a little bit closer in the family tree up to the modern groups, but not quite there yet. And specifically, we get vanid and Trianican turtles, things like Gamera Bana here, which I love B-movies, so that's a fantastic name. But what does that mean about this chunk of time between about 100 and about 84 million years ago, where we don't have really good sampling? That's a large chunk of time during an interesting period of time in North America, where obviously we're having some faunal turnover. We're going from that middle Cretaceous group, the organisms we see there, moving towards this late Cretaceous group, but we don't really know when it happened or how it happened. We just don't have a lot of fossils. Also, to sort of compound matters, this is what North America actually looked like during this period of time. the part of the continent was actually flooded by the Western Interior Seaway. So we had functionally two different continents. We had Laramidia to the west and Appalachia to the east. And whenever we do have fossils from this chunk of time, almost all of them are from Laramidia. So we have essentially nothing from Appalachia. And it makes it this very mysterious chunk of time, because obviously things were living here, we just don't know what they were, what they were doing. Which brings us to our, I guess, our great hope, the Woodbine Formation. It's a geological formation in East Texas, which makes it horribly convenient because, first of all, it is now on that Appalachian continent. It's a deltaic system, right near the edge of it, but it is Appalachia. And it's been dated to between 95 and 96 million years ago, which puts it in the Cenomanian. So that's right in that gap where we just don't have that many fossils. So again, here's that table as a reminder, and there's the Cenomanian. So all right, great, we do have this place we can go and rocks that we can look at and hopefully find some fossils to tell us what's going on. So what's the problem? We've known about the Woodbine Formation forever. Honestly, it's because the fossils are kind of rubbish. They're very rare, they tend to be pretty beat up, they're sort of scattered around. So this is actually a fantastic example of a croc fossil previously known from the Woodbine Formation. It's beautiful, it's like a handful of bones. It's a little frustrating, you want to go out there and you want to find this stuff, and the fossil record just isn't that great for this particular unit until the discovery of the Arlington Archosaur site. In 2003, two separate amateur collectors, amateur paleontologists, went to a construction site in the middle of Dallas-Fort Worth, just right in the smack middle of these suburban areas, found fossils, brought them to UT Arlington, said, hey guys, we think we have something important here, what's going on? It turns out that it is Woodbine Formation, except the fossils are beautiful, and there's tons of them. So this is just one shot of an area where we have a lot of different crop fossils. Let's see, we have leg bones over here, we have bones over here that have sculpturing and parts of the skull all jumbled together. Really beautiful preservation. To get a little bit larger site concept, this grid is by the meter. And while that might not be impressive to an invertebrate paleontologist who sometimes can go out and shovel up their specimens into buckets, for us, that's a really large number of fossils in a really small space. So what happened initially? As I said, these two collectors brought the fossils to UT Arlington, and at the time they had a student there named Derek May. Derek actually ended up picking up the research at this site as his Ph.D. dissertation. And right out of the gates, he immediately started involving these amateur collectors that were around in the area. They're the ones who found it, and he really wanted to keep them involved. In Texas, they're actually really proud of their fossils. They're really enthusiastic about it. They're excited when people find fossils, and they have a very active group of people who are in the Dallas Paleontological Society who wanted to be involved in the dig here. And it sort of blossomed into this whole group of people. They call themselves the Archosaurs. They named it the Arlington Archosaur site. And almost everything I'm going to be talking to you guys about for the rest of this whole presentation were found by these volunteers. So everywhere from high school students all the way up to retirees. We have one of the guys who actually found the site lives maybe half a mile away and has taken it on himself to, I guess, guard the site when people aren't there. So if we have to leave equipment behind, he'll go out and make sure no one's messing with it and things like that. So they're really invested in this site. They're really excited about it. You get these huge teams of people out from the communities who show up, volunteer their time and effort. They've undergone training to make sure they're collecting everything the right way. And all of this stuff gets packed up. And at the time, it was being sent to UT Arlington. I'll talk about what happened to it later. So Derek came in and he was very enthusiastic about citizen science. He really wanted to involve the community. He ended up having this huge, dedicated group of people who were all passionate about this site. And tragically, in 2013, Derek actually passed away. This was only about 2 years after I started working at the site and looking at some of the fossils. So we found ourselves in a situation where we were trying to kind of figure out how to keep the momentum going. We knew that Derek really wanted us to keep doing this, to keep working at the site. He had done an amazing thing by bringing these people together, these volunteers from the community, but none of us lived there, and it was pretty challenging. But his very close friend and longtime colleague in many of his papers, throughout his Ph.D. dissertation, and he was actually publishing before that, was a guy named Chris Noto from the University of Wisconsin. And Chris really took it on himself to keep the momentum going with this site. And part of that was he went back to people who had worked there previously. Myself, this is Thomas Adams from the Whit Museum in Texas, brought us in, and we have started trying to get together grant funding to try to keep this thing going. We've forged connections with actually the Perot Museum in Dallas, Texas. So all of this material, which it was really uncertain what was going to happen to it after Derek passed at UT Arlington, it's now housed in the Perot Museum. It's under care and curation there. And a lot of our volunteers have really taken this as sort of their way to honor Derek's memory. So we're, honestly, a lot of what we're doing right now is picking up projects that he had started and trying to keep them going. And there's an awful lot to talk about. There's an awful lot of cool stuff from this site, and we're happy to keep working on it in his memory. So what is the Arlington Archosaur site? What are we preserving here? Why is it so unusual? And what is it telling us about this chunk of time in the Cretaceous where we don't have a lot of fossils? Well, first of all, it is a deltaic system. So thinking about, say, the Mississippi River Delta is not a terrible point of comparison. We have a mix of different types of groups all on the same site. So marine things, freshwater things, terrestrial things that have all been deposited into these shallow sort of estuarine environments. We have a lot of plant material there that hasn't been studied in much detail yet. We're working on trying to get some paleobotanists out to the site. But what it does tell us, we actually have many lenses of charcoal. We probably had a lot of forest fires happening in this environment. The lenses of that, we have everything from pollen, so microfossils, all the way up to huge logs, large sections of these plants. And everything from vertebrate microfossils all the way up to very, very large crocs and dinosaurs and things like that. Now, it is a little tricky working with these because the fossils are beautiful. They're wonderfully preserved. They haven't been exposed long enough that the bones started to degrade, but this was a low energy environment. They were exposed long enough that they disarticulated. So we don't find these nice crocodiles like a Greek frieze, nice and spread out and articulated all together. We tend to find them jumbled up and spread all over the place. So you have this horrible jigsaw puzzle that you have to get them back together and figure out, okay, which of these bones actually belong to the same animal and which ones didn't, and who goes with who. So it's a big jumble, but we are being able to tease out quite a few interesting things. So I mentioned that we do have a mix of fossils from different environments. Ones along the top, we have sharks, sawfish, stingrays, things like that. From the marine side of things, we also have lungfish and some other freshwater fish groups. From the freshwater side of things, and then frogs and salamanders, and some mammals from the terrestrial input. Now, these are all sort of small organisms, and the microfossils are definitely something that we're looking into and we're expanding on. Probably most of these are new species. But the overwhelmingly dominant groups at the site are turtles and archosaurs. We'll talk about archosaurs in just a second. So turtles, we have at least 4 different species, probably more. Everything from very, very small ones up to great big, large. I mentioned those banded turtles. They have very, very thick shells. We have soft-shelled turtles. Turtle shell, as you might imagine, is pretty durable. It survives pretty well, so we have an awful lot of it at the site. And then archosaurs. It's right there in the name of the site, but sometimes an unfamiliar term for people. Archosaurs, or Archosauria, is the group that includes crocs and birds and everybody in between that's more closely related to them. So this includes things like dinosaurs. So crocs, this is actually the reason that I was brought in initially into this project, is we have some very large crocodilians here that are probably eating everyone else in the environment. This one was honestly why I was back in Arlington over the summer. We're sort of in an awkward position right now because the paper's out being reviewed. We are naming it. We're naming it Deltasuchus. The name was sort of leaked out by not us. So we're not supposed to use the name until it's formally published, but the cat's kind of out of the bag. This thing would have been 20 to 24 feet long in life. Those are the bones and the silhouette that we have of him. But to give you a little bit better idea of scale, This is a reconstruction of one of the individuals. We are missing quite a bit from this part of the skull, but there's the three of us in the background to give you an idea of how big this thing would have been. So this is just locked up in the Perot over the summer, looking at this stuff and trying to write a description. Now, I brought some examples of this because we've been trying to use 3D printing technology and 3D scanning for reconstructions to look at a lot of these fossils. These are 3D prints. The smaller piece is part of the prefrontal, so bone that's sort of the front part of the eye. The larger chunk is part of the nasal, so you can actually see the lip of the nose in this. But if we scan these things, we can actually do digital reconstructions like what you see here. If we have a bone from the right side but not the left side, you reflect it over, fill in the gap. So here's a reconstruction of what this critter probably looked like. And we had a paleo artist come in and graciously fill in the gaps.
Not very friendly.
So here's Deltasuchus. Probably was the top predator at the site, but once we really started digging into the collections, it rapidly became apparent that it was not the only croc there. We have at least 4 species. One is Deltasuchus. One is a slender-snouted guy, so called Terminaris. And then there's 2 other sort of mysterious ones that we don't have many fossils from. These are just a couple examples of some of the other croc materials. We'll talk a bit about both of this side of things. This is a piece of Deltasuchus. It's the chin right here. And this is also Deltasuchus. So we have a whole ontogenetic series from the species. We have little bitty babies all the way up to great big adults. We also, I'm going to be going back in January to try to tackle the next new species of croc. We have this really, honestly, kind of beat up, but interesting, very tantalizing little croc jaw. It only has 9 alveoli, so sockets where the teeth would have been in life, and that's odd. Usually crocs have a lot more teeth than that. They're also really large and kind of anvil-shaped, with these big round flat teeth in the back that we often associate with duraphagy, specializing in eating hard stuff. Dinosaurs, because everybody wants to hear about the dinosaurs. I love the crocs, but I understand. There was a species that was already known from the Woodbine before we discovered the Arlington Archosaur site. This is what we had of it, which was a little odd. Usually skulls are rare, but we had this thing's head. It was named Protohadros based off of that skull. After the discovery of the Arlington Archosaur site, we now have not the entire rest of the skeleton, but awfully close. And the colors are different individuals known from the site. So just a couple examples here. We have parts of the hip, parts of the jaw, parts of the leg bones, hands, vertebrae. We have smaller individuals, so we know what the juveniles were looking like. We have larger ones, so we can compare them to the adults, which is all awfully nice to know what the rest of this animal looked like. We also have lots of these theropods, these bipedal meat-eating dinosaurs. What's sort of fun is one of them is probably the earliest tyrannosauroid in North America. So not T-Rex, we're not there yet. T-Rex was a really big one all the way at the end of the Cretaceous, but a smaller, earlier member of this group. We also have a large Allosauriod, more closely related to the stuff we see from the middle of this, and a selection of smaller ones. We have very fragmentary records of these guys, but we know we have 2, 3, maybe a couple other small theropods from this site.
Stephanie, when you're saying small tyrannosauroid, how small? How big was it?
I did break off the scale bar. Okay, so that tooth, Tyrannosaur tooth,
6 inches?
we call them killer bananas because they look like bananas. They're actually even in cross section. So Tyrannosaur tooth with the root and all is about like that. These teeth, so still something I would not want to be chased by, but definitely not on the order of a T-Rex. You don't want to get caught by these guys. But honestly, you also don't want to get caught by the crocs. And what's interesting, we call it the Age of Dinosaurs. There's a good reason that we call it the Age of Dinosaurs. They were some of the top groups on land, but they were not the only things alive during the Age of Dinosaurs. And some of these things didn't particularly care that they were apparently supposed to be not as important as their dinosaur cousins, and ate them anyway. I love working on crocs. I love them, and I always kind of find it in my heart of hearts darkly amusing when I find dinosaur bones with croc bites on them, which is what this is right here. It's actually some of our Protohadros. That was apparently a snack for our Deltasuchus. The turtles have been absolutely worn out by this croc. We know this because you can actually look at modern crocodilian bite marks, and they have very characteristic features that set them apart from any other group. They don't have serrated teeth, so they don't leave striations like the theropods would have. And if you look at the spacing of the bite marks themselves, how big those teeth were, and if you get a bunch of teeth all hitting at the same time, you can line that up with the tooth row. We know that a lot of these bites were Deltasuchus. Deltasuchus was sort of ruling the roost in this environment, everything that ended up in the water.
Is that your artist's picture there?
Yes. We, there was a chunk of time when this paper on the bite marks came out in around 2012 and 13, where a lot of the science reporters who talk about paleontological finds were nicknaming animals, and you get bear croc and boar croc and super croc, and we just about died laughing because ours got nicknamed Nutcracker croc because we described some of the behavior that seemed to be preserved. They were flipping turtles up on their sides and cracking the shell that way. So it had been described previously in modern crocs as nut cracking behavior. So now I have this mental image of someone pulling the spine up of our crocodile to eat turtles, and it was just odd.
Did you
But
speculate why the teeth are so far apart?
It's the sockets are that far apart. It is kind of fun and odd. One of the things that sets this guy apart, you can see it in the artist reconstruction here, is we have two what we call pseudo-canines. Instead of having just one big tooth on the side, which a lot of these groups have, we have two right next to each other. So it is a pretty reasonable reconstruction of this animal, maybe slightly more slender snout. A lot of the artists' reconstructions were student artists at the time, but it was pretty good. This is the one that I guess Nat Geo commissioned to go out with the Nutcracker croc article.
So how do you know, you were showing that that image that had several different individuals and bones in there. How do you know for sure that it is that is the same species that you're looking at, and not, since obviously you found other variations? How do you know?
First of all, we have to be really conservative with our reconstruction of these guys, because as I mentioned, they're not all nicely articulated and put together. So for the animal that we're actually describing when we're naming the new species, it's all one individual, and it's pretty much the skull. First of all, post-crania crocs can be very conservative. If you cover up their heads, I'm not saying they look identical, but it can be a little confusing. So we have all this post-cranial material. We can't use it in the description because we can't make sure it belongs to this guy or Terminaneris, that other big croc that's present at the site, or maybe even someone else. But we have.
Is the bone chemistry the same among all the parts that you have?
Yes, preservation is very similar across the board on all of this. What we can do though with reconstructing the individuals and figuring out, okay, this is definitely one animal, the suture patterns, the way the bones actually fit together on the skull, it's like a puzzle piece. So if you take the right nasal and the left nasal from two different animals and you try to stick them together, it's not going to work. They're not going to fit. But if it is the same animal, they fit together perfectly, just like a puzzle piece, good to go. So everything in that skull actually articulates with a neighbor, so we have the connection points of the bones all the way around. So we're happy that this actually is one individual. We don't have this scary chimera of different animals all piled together and calling it one thing. How do we know that it was leaving the bite marks behind? The snout shapes of these different groups that we do have are wildly different. So Deltasuchus, big triangular snout, something like what we see in American alligators and Nile crocodiles. We often associate that with a generalist diet. They kind of eat whatever they want to. The little jaw, the one that we're going to be working on in January, big brown teeth, much blunter, rounder snout, probably eating hard-bodied stuff like mollusks. Terminaneris, very, very slender snout. Think something like a gharial, which they're not true fish eaters. They're fish specialists. That sometimes gets thrown around in the literature, but they're more small prey specialists. Then we have this other thing that's out there that it's tiny. Whatever it is, it's tiny. So we have these different groups, but their snouts don't look exactly the same. And if you can look at the way the tooth and the tooth row, how they're spaced, how far apart are they, how does that curvature of the jaw play out? We call them serial bite marks. So if you have a single biting event and multiple teeth hit at the same time, it gives you an idea of the shape of the snout. They line up beautifully with Deltasuchus. So not every single one of them, I'm not going to say every single bite mark in the whole site can be ascribed to this guy, but the ones that we can, particularly the very large bite marks, seem to be coming from this guy. and he is the dominant species at the site. The other ones are there, they're rare, but they also have different snout shapes, different snout sizes, different curvature of the jaw. So
As large as he was, 24 feet or whatever,
Mhm.
he was probably there a long time.
oh yeah, I mean, these guys, they do grow throughout their lives, and it does take them a while to get to be that big. So it was probably kind of old. We haven't done the histology on them yet. We only have a couple of bones that we could possibly slice open to look at. You basically get growth layers in bone of groups like this, just like, think tree rings. Not perfect, because you do have remodeling going on the inside, so as you're building new layers on the outside, it's eating the stuff on the inside, and it can be a little frustrating. But it is one way to get a handle on how old these things are. We have one beautiful humerus, and we haven't found it in our hearts to chop it into little pieces yet. So hopefully we'll find a couple others and not feel quite so bad about it in the future. But
Can't get MRI for the internal structure?
it doesn't show, the lags don't seem to show up in this kind of preservation. We're going to be CT scanning some turtles to look at pathologies, I guess, next week, actually. We're starting with modern ones, and then we'll have comparisons to the fossils. But the preservation sometimes makes it challenging to see the internal structure in that level of detail, unfortunately.
This is micro CT, right? That you
Mhm.
it still wouldn't be good enough.
We have done micro CT work with embedded teeth with bite marks before, and the trick with modern bone, it's not quite as dense, and you can see a lot more detail. But once you start the mineralization process and you get minerals in all those voids, a lot of that you have to physically cut it open and do thin sections. Which was a hilarious talk that I saw by a guy named Kevin Padian, where he was basically blackmailing a Russian museum against a United States museum to let him cut open holotypes to look at the histology. So he would go to the Russian museum and say, well, so-and-so in the US has let me cut up their holotypes. They're like, oh, well, please, go. And then he would go back to the one in the US and say, well, so-and-so in Russia let me do this. So he just managed to scam all these museums into letting him cut open these bones that we don't actually have that many specimens
That
of.
was Kevin Payne.
It's totally Kevin Payne. But it's great data, and we're really glad he did it, but he tells this story, and it's just like, that's sad and hilarious and incredibly on point about it.
Your tree ring analogy,
Yes.
what's the time spacing of this? Is it
We call them lines of arrested growth. The idea, the hope, is that they're annual. So the idea with trees is when it's warm and wet and awesome, you grow really
pretty
fast.
obvious,
And when it's cold and awful, you slow down, so you get these denser lines. The hope is every time you find one of these denser lines, that's basically the cold season. That's the crummy time of the year with animals like this that are very reliant on their environment for their metabolism. It's a little problematic sometimes. We have actually demonstrated that you still get lines of arrested growth in captives that are kept indoors in constant climate. So trying to figure out how it actually lines up with the biology is something that they're really fighting about right now.
So it's questionable.
And it always, no matter what, it's giving you a minimum age. Because as I said, you're putting new lines on the outside, but then the bone is remodeling on the inside and eating those earlier lines away. So you're always getting a minimum on this. And some bones seem to be better than others, I guess, to use. Some are going to be more heavily remodeled than others. But yeah, there's always caveats involved. So when we do this, it's always, here's the minimum age. So probably not the age, but it was at least X number of years old. Yes?
When you found the fossils in that construction site,
Mm-hmm.
what did you do about getting the fossils out, and for how long did you stop the project, and did you have any static about stopping?
If you wait a couple of slides,
Oh,
we'll talk
okay.
about where the
I'm
site's
sorry.
going in the future.
Go.
Because we are definitely in the conservation paleontology side of things. Right now we're just like, oh, get everything out because the subdivision is coming. Which, in all fairness, the people who are in charge of the site that own it and are developing it have been remarkably cool about the whole thing. They said, oh, okay, there's something important going on here. We're going to change up our plans. And we're going to start on this side of the property, but get on it. We're coming.
Does the antiquities legislation protect you there in terms of stopping things?
There are laws protecting fossils on public land.
That's right.
But private land, we don't have the same protection as, say, anthropological sites, as human sites. So it's viewed similarly to mineral resources. So if you find gold in your backyard, then good on you, it's yours. And that's their view on fossils as well. So there's not an extra layer of protection for that. Now, if you go and start stealing fossils off of like BLM land or national parks, then yeah, don't do that. There's laws in place to protect that, but it's on private land. So they didn't have to do this. So we're actually quite thankful that they didn't just mow over the whole thing and put a parking lot on top of it. Because they could have, legally, I mean, and it happens all the time. Luckily, these guys have given us some time to get the material out, and they're actually planning to build a park there, a community park. So it'll have interpretive signs, and we might get to keep working. The trick there, though, is all the agreements that we have right now are with the development company. The second they hand it over to the homeowners association, maybe they'll say, like, this is really cool. We have a dinosaur site in our backyard. That's fantastic. We want to take care of it. Or maybe they'll say, oh, dirty, paleontologists keep showing up in our backyard and running away with fossils, and they look kind of scary. So go away, never come back. We don't know what's going to happen. And that's why, honestly, things like the National Geographic and the Experiment.com grants we did this last year, it's like, we're running out of time, and we need to get as much stuff out as we can before we potentially don't have access to the site anymore. I have to put in slides of poop. Yep, that's exactly what this is. I mentioned it's a low energy environment. You've just been sitting here staring at poop this whole time. You didn't even know. So, we have a lot of coprolites here. So again, it's low energy. You're not going to get these preserved if it was a high energy environment. The water's bashing everything around. And they're often really hard to line up with exactly who was leaving them behind. So we can usually only get them down to sort of general classifications based on size, composition, and shape. So we do have some dinosaur coprolites. We have sharks and fish. We have this sort of mysterious, we're just calling it reptile at this point. And again, why do we think that the crocs were sort of the top dog here? A lot of croc coprolites from this site. So whatever they were doing, they were eating a lot of stuff, and they were hanging out for extended periods of time afterwards. So the diversity of the Arlington Archosaur site, why is this important? What have we found? So we have first record of a large allosauriid and first tyrannosauroid on Appalachia. So remember that eastern continent that we don't know much about. We have a large dromaeosaur of some kind and at least 4 more other small theropods. So that's the meat eater side of things. We have at least 4 species of crocodiliform. 2 of them, at the bare minimum, are new species that we've already submitted one. We're going to be submitting the other one, hopefully, sometime in the spring. Lots of new species of microvertebrates, so all of those little fish and frogs and stuff like that I showed you before are all in the works, getting researched. Paleobotany, we know we have stuff there. We know it's interesting. None of us are paleobotanists. We're sort of on the hunt for a specialist to come in and help us out with that.
In the third bullet from the bottom, what do you do when you find a new species about going back and trying to find out where the origin was, or what the origin age was?
Well, the age of the site has the age of the Woodbine Formation. Something that a lot of people don't always realize is you immediately think radiometric dating. Radioactive isotopes, the decay is at irregular rates. We can go in and figure out how old it is. That is awesome if you're looking at igneous rocks. So you've had molten material, it's cooled down. That's when the clock starts. Fossils don't do so great in molten material. So what we actually end up doing is very complex correlations. So you have an ash bed over here, maybe at the top it tells you age X, you have another one here, it's age Y, so everything in between is between X and Y. What fossils do you find here? Okay, do we find the same fossils over here? We're in that same time frame, even though we don't have the rocks to sample for the date over here. So for this site, a lot of the invertebrates, the marine input, we call them index fossils, and we know these things are only around during this slice of time. And that's how we figured out how old it is. So the fact that it was found in that same layer with these index fossils, we aren't having to do any further testing on the bone itself. We know it came from that same window because of everybody that's in there as a neighbor, I guess you could say, in its site. Is that what you were asking, or?
Yeah.
Okay, yeah.
Good enough.
It sometimes even surprises people in the geology department. They don't realize how reliant on index fossils the geologic timescale really is. We talk about the radiometric dating serves as anchor points. Those are our real dates.
Well,
These are our
standard.
times. But correlations through time, a lot of times it's based on fossils, which is why it sometimes gets a little bit challenging. The American Southwest, in the age of dinosaurs, we don't have a lot of nice igneous beds out there to get those hard dates off of. So there's a lot of question about some of these early Triassics, first part of the age of dinosaurs. A lot of the dates there are really kind of fuzzy just because we don't have anything to compare them to. So it's a problem, and it's why we still like our field mappers. We still love our geologists going out there and finding this stuff because it needs to be done. I talked about this gap in our knowledge at the very beginning, so why is this important, and then to unpack a little bit of what we found so far. So the early Cretaceous, this Aptian Albian window, there's our list of what we have. Late Cretaceous is the exact same slides I walked through bullet point before. What does this do for the Woodbine? As you might expect, we have a nice mix between the two. We're seeing this transition from the early Cretaceous to the late Cretaceous and how exactly that happened. We actually do have one site from this east-west problem, this Appalachia versus Laramidia. So again, as a reminder, yellow is us, that's our Appalachian continent. Muscontucket number in Utah it's a little bit older, so it bridges that well-sampled Albian, but then goes into the poorly sampled Cenomanian. What's interesting is there are a lot of similarities here. So you have the same big groups. So you might have, say, a type of Phallidosaur crocodiliform on both Laramidia and on Appalachia, but the species are not quite the same. Which makes an awful lot of sense, because if you started with one landmass and then the interior flooded and separated it out into two land masses, that's sort of classic. You know, each population is now separated from each other. They go off on their own evolutionary trajectories. This pattern is really, really nice when it comes to all the terrestrial stuff, dinosaurs, your turtles, things like that. Doesn't work quite as well when we get to the marine component, obviously, again, because they can swim back and forth and they don't care. But yeah, it's still a nice point of comparison that, okay, we should be finding a lot more of this stuff in Appalachia that was around in Laramidia, just preservation-wise and perhaps lack of luck in finding stuff-wise, we haven't yet. So you were asking about the future of the site. Here's the cool thing and also the horrible thing about the site. This is what it looks like if you're standing there staring at, you know, this dark part at the bottom is where all the fossils are coming from. Doesn't look terribly exciting, right? What do you see when you turn around 180 degrees from that? The Dallas Cowboys football stadium. So there is no way that this property is not going to be developed. And okay, it's not that close, but still, you can see it. It's right there. As a field geologist, it's awfully nice, because usually you think of doing the paleontology and you're out in the middle of nowhere and you have to pack all of your food in. And here we're like, oh, it's hot. It's 1 in the afternoon in Dallas. Let's go to Subway, which is horribly convenient. But on the other hand, no, really, the construction crews are coming. We've got to get this stuff out.
But you found that about 30 feet below original ground surface. Is that right?
Yeah, so that face, it ranges from 15 feet-ish high at its lowest to about 30. And as a paleontologist, you just look at it, and then you look at the subdivisions that are on the top part, and it's like, okay, I can't just go knock them all down and dig up this whole area, as much as I might want to. Because there's clearly fossils under there, but that's Dallas. I mean, that's the entire area. It's very fossil-rich. It's fossiliferous, as we were discussing. But this site, it's such a great site, but we haven't known about it for that long. As I mentioned, it was only discovered in 2003. Really, Derek's research on it didn't get started until 2005. So we haven't had that long in comparison to something like the Burgess Shale in Canada, where we've had over 100 years of paleontologists stomping over it and finding everything they can. So the development company, as I mentioned, has been very cool. And they've said, okay, we're going to leave you alone for as long as we can manage. Get this stuff out. They're talking about making this park. So for what it is, we're actually very thankful for the time that we've had. But we're really trying to amp up the research of the site because we have to get it out of here. So it's one of the reasons there haven't been as many publications yet, and we have this backlog of everything that needs to be described, is because a lot of the focus so far is just get it out of the ground. But we are starting to. So we have the description of the bite marks out. We have the description of one of the new species of crop. We've redescribed Protahadros. I mean, there's all kinds of stuff that's starting to come out now that we've known needed to be written, but the focus is just now shifting there because the site's about to be, it's about to shift out of control of the development
So
companies.
you've had 11 years, what would you project to be if you, to be able to get, the
Oh.
lion's share
We
or the crocodile share of what you're
We have 2 to 3 years, the crocodile share, sorry, 2 to 3 years before this is done and it's handed over to the homeowners association. Will we get everything out? Absolutely not. It's difficult to say though, because as I mentioned, usually the Woodbine Formation, not very fossil rich. So we aren't sure how far this lens of really fossil rich material extends off in the hillside. And to be honest, even if I had 1,000 years to dig this up, you're eventually going to start running into people's houses, and they get cranky if you undercut their foundation. So we can't do that. As a point of comparison, the Gray Fossil site here in Tennessee, they've said it's a very fossil-rich site. It's physically smaller as a sinkhole, but vertical digging, they said they could dig that thing for a couple hundred years and still be pulling fossils out. So again, in a wonderful world, as a paleontologist, I'm like, clearly everyone has to move and we get to play in the sandbox forever. Realistically, that doesn't happen, so it's one of those things where we're doing the best we can. But we're also being sort of interested in the fact that this is Woodbine. And the Woodbine is constantly being described as kind of rubbish. And you can't find, everything just looks awful there, it's all scattered around. Well, what is this? What is this doing here? And is it unique? Is this like the one time in all of history that we get decent preservation for the Woodbine? Or if we revisit some of these other sites, are we going to find these lenses of fossil-rich material if we just look a little bit more closely? That's sort of our long-term plan. After we can't work here anymore, we're wanting to start visiting some of these Woodbine sites to see, have we overlooked the diversity here? Do we possibly have better fossil preservation than we thought? I don't know. I can't answer that. We're hopeful. It's very rare that you find something like this that it really is the absolute only one that's out there. And the Woodbine outcrops all over Texas. So we're
This
going
is
to
kind of a swamp area, it seems like.
So estuarine, deltaic, sometime, water comes up, it's a little deeper, sometimes it's a little more shallow. So it would fluctuate some, but when all of this was happening, it was all deltaic. I think.
Yeah, any ideas on how the animals died?
Honestly, we don't have any really obvious other than some of the turtles who clearly died by way of being lunch. A lot of these, you just have a long enough period of time, things are just going to die of disease and old age. It wasn't like a single catastrophic event. This is a long period of time where these animals were just living and dying in the environment as they would, and it's sort of piled up in this depositional system. So we don't have really, I can't tell you, like that big skull, I can't tell you why that thing died. You can't always tell. It's sometimes very difficult to say in the fossil record. So unless they have big croc bites all over them, and then, well, maybe that's what happened. But then again, maybe it was scavenging. I try to not be super definitive on this stuff because at the end of the day, I can't be. So I'm sorry about that. Palynology, the plants really need to be worked on. So Noto was actually talking to some people at Geological Society of America and trying to find someone who might be interested to come in, because all of the local people who work on palynology, it's all Asian mammals, it's all more recent stuff. So we need someone who likes older things to come in and play with our toys.
This would be freshwater, though?
Mix.
Yeah.
So you have the freshwater input from the river, you have it dumping into the ocean. So again, you have fluctuations where sometimes it's saltier, sometimes it's fresher, sometimes it's kind of in the middle. This has been an interesting project in terms of funding. The larger proposal that you mentioned in my introduction is citizen science research, and it's a department project that's going towards recruiting undergraduates. And we're definitely trying to get more undergraduate research on those projects we're folding this into. We got funding from the National Geographic Foundation, from actually their conservation side of things, which is usually for living animals. And they said, well, it needs to be conserved, so we got funding from them for that. But the funding that we got first was actually through experiment.com, and this is sort of a weird, brave new world of funding, I guess you could say. Experiment.com is crowdsourcing. Basically, it's a site where you get online, you say, I have a cool project, let me tell you about my cool project. And total strangers, through the magic of the internet, show up and give you money. Which is great, and it's actually really flattering. It's like people I have never met, who probably, maybe never even go to Texas, think this is cool enough that they just gave me $20. And, we
So
get enough donors.
You're generating crowdfunding.
It is, yeah. And our initial funding to sort of kick things off the last couple of years, it was crowdfunding. Okay. And we've since gotten, I guess, more traditional sources of grant funding for it. But we had about 120, just mostly total strangers, donate to the site. So we go in periodically and we have updates and we say, okay, people who gave us money, here's what we've been up to, here's some pictures, here's what we found. And it's sort of an interesting new platform for getting a kernel of funding, just kick things off, and then maybe later on using that pilot study to get the more traditional sources of funding. And Derek definitely kicked things off with really involving citizen scientists, and we're trying to keep that ball rolling. We're trying to bring in more people and train them at the site, but we're also starting to get a little bit more creative with our outreach to get it just beyond Texas. This is just an example. This last year we did a Reddit Ask Me Anything, which is horrifying. It's like your comps all over again, except your committee is the Earth. Because absolutely anyone can just get online and ask you whatever, thus the name, Ask Me Anything. And we asked them to actually send us some information. When we did, and it was just a couple of hours, and we were online just furiously answering questions. 10 million people saw it on Reddit's page. 37,000 people actually clicked through and started reading the answers that we were writing. And 6,000 people thought that it was cool enough to actually react. Get in there and say, oh, that was sort of
In just
fun.
2 hours.
Yeah. It was 2 hours of us, and then there were a couple afterwards we were like, people kept asking, and so you have to kind of back off, because if you just keep answering, it can go on indefinitely.
What time of day was it?
This was, I think we actually aimed for sort of mid-afternoon, so we got to eat lunch, get to refuel, and then sit at the computer and it was everything from really, really technical discussions of things like, how do you know that this thing was all the same animal? How do you actually piece it together? All the way to, when did you know you wanted to be a paleontologist? Or, it was the whole spread, which is a lot of fun. It is kind of intimidating. It's just because anybody can show up and ask. And we had moderators, so if there was anything where they were being, we'll say, offensive or anything like that, then we didn't have to deal with that, thankfully. But yeah, and people will do this. There's the subreddit that we were on was called Ask Science, and all they do, the entire point of the page is they get researchers from different institutions to come in and just agree to answer questions for anyone. And it was amazing. We got that level of feedback. We were on the front page of Reddit, and people in the department are like, do you know that you're on the front page of Reddit? Because that's weird. It's like, don't talk to me, I'm answering questions. It's scary. Another thing, and this is of sort of local interest. As a paleontologist, people show up at our department all the time with fossils. And 9 times out of 10, you feel really bad because they're very excited and it's not a fossil. Sorry, I'm getting dry mouth. A couple months ago, a guy showed up at our department and said he had dinosaur fossils, and Colin Summerall and I went, and we walked down and we shook his hand and he started pulling out trays, and they were dinosaur fossils from West Tennessee. If you guys have ever been up to the McClung Museum, they have the one non-avian Tennessee dinosaur that's on display up there, and we're pretty certain this guy has rediscovered the site. So we're going to be going out in December to look at this dinosaur material from Tennessee.